Hi, this is Dr. Bailey, and in this video, we're going to look at some past IB exam paper questions related to topic 5.4, which is all about cladistics. So here we have the first question. It says, using a cladogram, identify one diagnostic feature that characterizes the given groups of vertebrates A, B, and C. So we're talking about A, B, and C. What is the trait that goes with them? Now, if you remember the cladistics, it means if you are coming here, this is basically where you have an ancestor, right? And this ancestor, that means they all have in common all the traits, right? The common traits, they all have the same. However, if you don't have a trait, you're going to have to move out of that group. So in this case, the fish, one thing about the fish is that they have gills, right? So they move out of the group while the rest continue. That means birds and mammals continue. Now, what is it that the birds and mammals have that the fish don't have? And here you got to think of an idea that has the birds and the mammals. We'll see, maybe we can talk about the homologous structures, their bones, right? It can be that. Structure. You can talk about the 5 fingers, right? And then here, what does the mammal have that the birds don't have? I can't say the placenta. That would be 1. So let's look at the mark scheme. So first the gills, as I said. A, endotherm, warm-blooded. Ooh, I could have used the warm-blooded. Oh, the pendactyl, which I mentioned here, right? The 5 fingers, right? And the hair, the fur, the mammary glands, or the milk. And what have I mentioned? The placenta. Didn't have the placenta here. So I would not get the point on the last one. But hair, fur, mammary glands, milk, now that I know, I am ready to get that point. So this is how we go through. This is why I say it's always important to go through previous IB questions, because there is where you get more ideas of answers they can get. So we also can see here that they are endothermic, warm-blooded. You can see that they had lungs. You can also they had the forelimbs. There's so many other ideas. And the more you see it, you get more ideas to put in the test. That's why I always tell students, always go back and see many of the questions of the previous IB exams, even though they're not going to repeat the questions. But the mark scheme, there's a lot of information there that you can use it again. So moving on to the next question, describe what is shown in a cladogram. Now, most important thing when they say that is define what cladogram is. And cladogram is basically, it's a type of map, you will say, in which shows species placed according, this is me putting my own words, species placed according to their similar traits, right? Trait. So here you can see their relationship. That means how close they are to each other. Here you can talk about the ancestors, if their ancestors are common or not. Here we can show, we can create a cladogram by showing their DNA and protein, how close their proteins and DNAs are. So here we're just showing also divergence, right? What I mean by divergence, that means they had a common ancestor and then eventually they will split into new species, species number 1 or species number 2. Now that split happens, that will be known as divergence, because somehow a mutation has happened, right? That cause a DNA to change and eventually protein will change. But if you go back, they all had a common ancestor. So that's what cladogram is about. Now let's see the mark scheme. So the similarity differences between organisms, species, or clade. So you can see here as showing place according to their similar traits. So I get that point. Evolutionary relationship, closeness, common ancestors. So you can see here I'm talking about them having common ancestor. Divergence, split, speciation. Okay, so here I show the divergence, right? Which basically here, right here, the node where there is a split that happens. What else we have? Relative similar differences between base sequence and amino acid sequence. And I did mention here, again, how we can look at their DNAs and proteins and see how close they are or not. So Out of these guys, you can see that this is what cladogram is about. So whenever you're going to answer a question about cladogram, most important, talk about how they have different and similar traits. Second, talk about them having a common ancestor. Third, talk about divergence, where they start branching. That's the whole point about branching. And then last but not least, talk about you can use DNA and amino acid in order to do those branchings. So these are the 5 things that you must mention about cladogram if they ask you. The next time they ask you, mention those 5. Now, list 2 types of evidence to determine the species belong in the same clade. So again, it's asking for 2 types of evidence to make the clades. Obviously, that will be DNA and protein or amino acids. These are the 2 evidence that you can use. Let's see. DNA and amino acid. So you can easily see that all the clades can be done according to these 2. That's obvious. You know it from now, DNA and amino acid. Next, the following cladogram shows 3 possible evolutionary routes for turtles 1, 2, and 3. Okay? The taxa in italics are extinct, so that means this section here, they all have gone, right? They're extinct. And then state the organism most closely related to the lizards. So searching the lizards, I have to find what is most related. Now here it means that they have a common ancestor, so that means they kind of just did a recent split into Tuatara. So Tuatara here is the most close related. As you can see, they both have a very common ancestors in this case. So let's see the answer here. Yes, it's Tuatara. Now, let's go for the next one. It says, based on the taxa shown, deduce the difficulty in gathering the data to study the turtle ancestry. Well, if these guys have been extinct, it's very hard for you to find their DNA or their proteins, so you don't really know if this was the right pattern, right? That's the first thing. So once again, because they were extinct, hard to have DNA or amino acid evidence. That will be the first thing I'll write, right? The second thing that we can see here is that we kind of probably made mistakes about the turtle 3 here, since we don't have enough information. So turtle 3 here, it's kind of vague in the way we put in the clade. So I would say turtle 3 is not 100% accurate. And we don't have enough data to do it. That will be one of the reasons why I think it's very difficult to gather the information. So let's see what they have for mark scheme. Some taxers are extinct or convergence. Oh, I didn't think about that one. Convergence would be that maybe some of these guys joined to be together. Like you see this one here, you don't know if the turtle 2 came out from joining these 2 or something that was joined before. So you don't have the exact data. You don't have the information like DNA, amino acids to make sure was it combined. But just the fact that I said that it's instinct, that's just 1 point, because the word says or, so it could be this or that. So this was a 1 point, and I still got the point because I don't have the DNA from the instinct animals. Moving on, it says outline, and outline is basically, again, it's bullet points, types of evidence that can be used to place the species in a particular clade. This goes back to the information. The evidence we have is DNA as well as the amino acid. And then, of course, we got to look at their structures, right? Such as the fossils. We can also look at their fossils and see the way their structure is in order to place them. And, of course, we got to look for a common ancestor. You know, the closer they are to each other, that means the closer their common ancestor is, right? So if 2 organisms are very similar, that means automatically, most likely, that they have a common ancestor that they just diverge from. So let's look what they have here. A clade, okay, obviously always start with the definition. It's a group of organisms that have evolved from one common ancestor. I did mention the common ancestor. Identify the base sequences of the genes. So I did mention using DNA. Identify the amino acid. I had mentioned talking about using amino acids. Comparing homologous structures. I did talk about that, like looking at their bone structures. Fewer differences. closer they diverge from a common ancestor. I did mention that, right? When I mentioned here, in this point here, two organisms who are very close and most likely they come from the common ancestors. But I could have said is showing the clade how 1 and 2, they're very related and they have barely any difference between them is because they probably just diverge. They just diverge from that common ancestor right here. So going back here, we have basically put the most common things they can talk about clades. So first of all, and the most important of all, mention what a clade is. It's just a group of organisms that might have some differences or similarities of the traits coming from that common ancestor. So we're looking at their differences and the similarities, not just the similarities. Remember, you're looking at both differences and similarities of the traits. Then as evidence, we use DNA, which is base sequence, or amino acids. These are two ways that we can do it. We also can look at the homologous structures. That means in their bone structures, see how similar it is. And if they have less than some few differences, that means they most likely just diverge from a common ancestor. Now, moving to the next question. Here it says, cladistics and other evolutionary evidence suggests that mammals and birds have more recent common ancestor than mammals and amphibians. That already says, that gave you information that said bird and mammals. They're much closer, right? They're much closer than the mammals with the amphibians. So the amphibians are going to be a little bit more far, I would say that. And then draw a cladogram to show the relationship between the mammals, birds, and amphibians. So if it's more recent, I wouldn't draw this way. I would draw the other way. I would draw the opposite. I would first put the amphibians, and then I will put the birds, and then I will put the mammals. Why? Because they're very close related, and I wouldn't do the opposite way, because if I do this way, that means the amphibian somehow kind of had some new idea, some new trait that these guys didn't have. And these guys, usually since they're recently, they should be more to the new part, and the amphibian should be more to the old part. So I would use, as highlighted, this would be the cladogram that I would use. And I would definitely not use this one at all. So let's look at what they have here. Now, the glitch shows evolutionary relationship through a common ancestor, which we did. We showed the common ancestor here between mammals and birds. Cladistics use DNA protein derived. We have DNA. Time of divergence is related to the number of differences between the DNA and the protein. Homologous traits are used to place the organism. So here we're just saying the information about clade. It didn't say anything about talking about it, but somehow it's written here, draw and talk about it. Show more shared characters means more recent common ancestors. Look at here. More shared traits means more recent common ancestors. So they're most likely to be together. So that's why you can see that when I put here the mammals and the birds, they're much common ancestors than you will have the amphibians right here. So here is 3 possibilities of how you want to draw this. So as I was drawing this, I used this one right here, but you can also do the other way. You can have them start here, which the amphibians came, and then the birds, and then the mammals. Or you can have this way, which the amphibians separated, and then there was a separation between those two. But of course, right here, it shows it as a common ancestor. That means they are much similar to each other and less differences compared to the mammals with the amphibians. So these are 2 ways in which you can do the cladogram. Last, outline the use of analogous and homologous traits in natural classification. So basically, what are the differences? So we have here analogous, and here we have the homologous. So we're going to look at the differences. So when I say equal, it means similar. When I say difference, when I do this, it means difference, right? So this is similar. What do they have the similar? They have the similar function. Differences, they have different structure. And homologous is completely the opposite. It's similar structure and different function. This one is more of a convergence, and this one is more of a divergence. This one has no common ancestor. These ones, they do have common ancestor. What else can I think here that I could put? I can give examples. I can say here, like, for example, the dolphin and the shark, right? They both swim, but the dolphins are mammals and the shark is fish. Completely different groups. Common ancestor. In this case, I can talk about the pentadactyl, the fingers, right? The 5 fingers, and I can talk about how humans and bats, they have the same bone structure. However, humans would say that they walk, right? And the bats usually fly. So here I have put all the differences between the two. So let's look at the mark scheme. Here, analogous, different evolutionary origin, which that means they do not share the same common ancestor. That's what it means. When I say different origin, that means not the same common ancestor. So it's a convergent evolution, and that means their base analog strings brings together the similar species. That means they were different. The similar species means they were different species, but they come together to form a specific function. Homologous, on the one case, they have similar structures, and we talk about pentadactyl limbs, but they have different functions. This is adaptive or divergent evolutions. You can talk about homologous traits. In this case, you can talk about the pentadactyl, and these homologous has predictive values, matches evolutionary history. In this case, you can also say they have a common ancestor, which is right here. Homologous trait has predictive values, matches. That means they have a common evolutionary history. That means their history is very common and similar. So this is the difference they can get. So basically, my little sum up here has nailed all these points. But of course, you have put in the words. By putting them in the words, into sentence, you can have a very strong base. So if you have this in your mind as an outline in your head, then you place them into sentences in your test, then you got through. So once again, analogous and homologous, similar functions, different. Similar functions for analogous, a similar structure for the homologous. This is different structures, this was different functions. Convergence, divergence, no common ancestors, it has common ancestors. An example, dolphins and the sharks, and here we have the humans and bats, which they have the same bone, which is like the pentadactyl finger, but one is walking, one is flying. You have that in your head, and you write these guys, you will always nail all the points you can about these two. And that's pretty much it for 5.4. And here are some extra things they might ask about defining cladogram and node, or they will give you another cladogram and you have to find the information, such as the primate ones, like the humans with other monkeys and gorillas. And the reason, and here is that one of the things is application, but I haven't seen a question comes like this in the test yet, which is the reason and the evidence for a reclassification of figwort family. You have to know this information. So it's in my video. You really have to know how they reclassified the cladogram. And again, it's all about finding technology and the DNA. And this is pretty much it. Thank you.